My name is Joshua Mugabi. I welcome you to topic 4, respiration. Our learning outcome B. Analyze the biochemical processes leading to ATP production in living organisms and how these processes are affected by physical activities and respiratory poison like cyanide. Details of biochemistry are not required. We are going to look at the learning activity A. In groups, learners use simulation that demonstrate the process of glycolysis, focusing on its stages, key molecules involved, and its significance in cellular respiration. Make a write-up and present it to the class plenary. Glycolysis in cellular respiration. Item 1. We are going to look at energy release without oxygen. A student runs a short sprint and experiences muscle fatigue even before breathing heavily. The muscles are still producing energy. Task. Define glycolysis. State where glycolysis occurs in the cell. Explain why glycolysis can occur in the absence of oxygen. Define glycolysis. Glycolysis is the series of enzyme-controlled reactions in which glucose is broken down into two molecules of pyruvate with the release of energy. State where glycolysis occurs in the cell. Glycolysis occurs in the cytoplasm, cytosol of the cell. Explain why glycolysis can occur in the absence of oxygen. Glycolysis does not require oxygen. All reactions are enzyme controlled and occur in the cytoplasm. We are still on glycolysis in cellular respiration, item 2. Let's look at the stages of glycolysis. Learners examine a flow chart showing glucose being converted into pyruvate. Task. Outline the main stages of glycolysis. Name the key molecules involved at each stage. Outline the main stages of glycolysis. Stage 1, phosphorylation, or what we call energy investment phase. Stage 2, splitting, what we call lysis stage. And stage 3, oxidation and ATP formation, energy pair of phase. Name the key molecules involved at each stage. Stage 1, phosphorylation or energy investment phase. Glucose, a 6-carbon, is phosphorylated using 2 ATP and it forms fructose 1,6-biphosphate. Stage 2, splitting, lysis stage. Fructose 1,6-biphosphate splits into 2,3-carbon triose phosphates. Stage 3, oxidation and ATP formation, energy pair of phase. Triose phosphate are oxidized. Oxidized NAD is reduced to NADH. ATP is formed by substrate level phosphorylation. The final product are 2 molecules of pyruvate. Glycolysis in cellular respiration, item 3. Let's look at key molecules in glycolysis. A teacher asks learners to identify substances that gain or lose energy during glycolysis. Task: list the key molecules involved in glycolysis. State the role of each molecule. List the key molecules involved in glycolysis. We have glucose, ATP, ADP plus inorganic phosphate, oxidized NAD, reduced NAD, NADH, and pyruvate. State the role of each molecule. Molecule glucose is the initial substrate. ATP provides energy in the early stages. ADP plus inorganic phosphate used to form ATP. Oxidized NAD, it is the hydrogen electron acceptor. NADH reduced carries hydrogen to mitochondria. Pyruvate is the end product of glycolysis. Glycolysis in cellular respiration, item 4. Let's look at energy yield from glycolysis. Learners calculate the ATP gain from glycolysis. Task: state the total ATP used in glycolysis. State the total ATP produced. Calculate the net ATP gain. State the total ATP used in glycolysis. 2 ATP used during phosphorylation. State the total ATP produced. 4 ATP produced during substrate level phosphorylation. Calculate the net ATP gain. Net ATP gain equals to 2 ATP per glucose molecule. Glycolysis in cellular respiration, item 5. Let's look at the significance of glycolysis. A yeast cell and a human muscle cell both carry out glycolysis. Task: state the significance of glycolysis in cellular respiration. Explain its importance in anaerobic rest conditions. State the significance of glycolysis in cellular respiration. Glycolysis is the first stage of respiration. It provides pyruvate for aerobic respiration, produces ATP quickly. Explain its importance in anaerobic conditions. Allows ATP production without oxygen. Essential in anaerobic respiration. For example, muscles and yeast. Glycolysis in cellular respiration, item 6. Examination style application. A student states, without glycolysis, cells would not survive. Task: justify the statement. State one consequence if glycolysis is inhibited. Justify the statement. Glycolysis provides immediate ATP. Supplies intermediate for other metabolic pathways and occurs in all living cells. State one consequence if glycolysis is inhibited. No ATP production in anaerobic conditions. Cell death due to energy deficiency. Marking emphasis. Correctly define glycolysis. Outline stages in correct sequence. Identify key molecules and their roles. State ATP yield accurately. Explain biological significance. Summary. Glycolysis is a cytoplasmic anaerobic pathway in which glucose is broken down into pyruvate through enzyme-controlled reactions. It involves phosphorylation, splitting, and oxidation stages, yielding a net gain of 2 ATP molecules and reduced NAD, making it vital for cellular respiration. Thank you for participating in topic 4 respiration. Our learning outcome B, analyze the biochemical processes leading to ATP production in a living organism and how these processes are affected by physical activity and respiratory poison like cyanide. Details of biochemistry are not required. We have been looking at learning activity A. In groups, learners use a simulation that demonstrates the process of glycolysis, focusing on its stages, key molecules involved, and its significance in cellular respiration. Make a write-up and present it into the class plenary. If this has been useful to you, visit me at Mugabi Joshua Biology on YouTube.